Okay, so hello, I am Vera Aldeias. I am a researcher and a current coordinator of the Prehistoric Coastal Adaptations Research Group here at ICArEHB, and I have the pleasure of convening today's session of ICArEHB Dialogues. It is my pleasure to introduce you to two distinguished researchers who focus on tackling the question of when did past humans started to explore the coast and its resources. As usual, we will first listen to the presentations that our speakers prepared, and then we will have an open time for many voices dialogue. So if you have questions, I suggest that you write them down in the chat room, and then I will call your name so you'll be able to turn on your microphone and ask your question directly. Alternatively, during the discussion, you can also raise your virtual hand and let me know that you want to intervene. Our speakers today are Klervia Jaouen, our guest researcher, and Erich Fischer, our ICArEHB speaker. What I personally find particularly exciting in having a dialogue between Klervia and Erich is that they come from distinct academic backgrounds and take different but complementary angles to answer the question of origins of coastal adaptations in human history. Before we listen to their talks, I want to briefly present to you Klervia and Erich. I first met Klervia in 2013 when we were doing our postdoc in the Department of Human Evolution at the Max Planck Institute for Evolutionary Anthropology in Leipzig. Klervia is a geochemist by training, and she was awarded a PhD from University of Lyon in 2012. Today, Klervia remains an associated member of the MPI in Leipzig and is now also the principal investigator of an ERC starting grant at the University of Toulouse. Klervia's ERC aims to use non-traditional isotopes to track the consumption of marine diets in ancient hominids. Klervia is a world reference in the application of isotopes, namely the so-called non-traditional isotopes, to decipher trophic food chains and past human diets. It is a pleasure to have you here with us, Klervia, talking about your research and dialoguing with Erich, who I will like to introduce now. My first contact with the research of Erich Fischer was in the early 2000s, when I heard him give a talk on the archaeology of Southern Africa. I remember being very impressed, not only with the scientific content of Erich's presentation, but also his astonishingly slick images and photographs that really transport us to the archaeological sites. Erich is an archaeologist and was awarded the PhD dissertation in 2010 from the Department of Anthropology at the Arizona University of Florida. Erich has been an assistant research scientist at the Institute of Human Origins at Arizona State University and an adjunct faculty member at Glendale Community College. And we are very happy to announce that Erich is joining us here at ICArEHB, and we are all looking forward to meeting him personally as soon as the Portuguese authorities actually let him into the country. Erich has extensive work in Ethiopian and South African archaeological contexts, namely at the well-known cave sites of Pinnacle Point. Since 2011, Erich is the PI of a large interdisciplinary project, the P5 project, in Pondoland in South Africa. Erich's research focuses on coastal adaptations, coastal foraging, and their role in the evolution of early modern humans. Finally, Erich has also extensive publications on documenting archaeological stratigraphies and modeling in an overall approach that he coined as archaeoinformatics. So it is a pleasure to have Erich talk to us today about the origins of coastal adaptations. So without further ado, I can pass the stage first to Klervia and then to Erich.
For this invitation. I'm really happy today to be able to contribute to those dialogue seminars and to be able to talk about isotopes and what they can teach us about coastal adaptation of past populations. So maybe the first question that you are going to ask is, why is it interesting to know if ancient humans were exploiting marine resources? So I'm sure it's a question that Erich Fischer, who's going to talk just after me, will be able to answer in more details and maybe in a more interesting way. But I just wanted to give you some insight of why, as a geochemist, I got interested in that question. Why did I what did I understand basically about it? And basically, why it's interesting to know specifically if ancient humans ate marine food products is basically because marine food products are a source of important nutrients for the brain development. And I think in particular about the omega-3, some type of omega-3 that are pretty in a pretty high concentration in the brain, but that are not so easy to find in other food products. So very interesting resource when you want somehow to grow bigger brain over the course of evolution. So of course, nowadays you can have a big brain and not eat fish because our diet is diverse and we eat like we have a lot of food, we eat sufficiently, but in the past maybe it could have played a role. It's also a stable resource. Wherever the climate is, you can always fish. So this is also in an adaptative kind of way very interesting. And not to talk about biological evolution, but more like on a cultural evolutionary perspective, it has been shown that fishing impacts the mobility, of course, but is also usually associated to a more complex hierarchy and a more complex technology, as well as higher demography. So it can have many impacts on societies, on human evolution, basically this adaptation to the coast. But the question now is, when did it happen? So we don't exactly know, but maybe Erich is going to give us some insight about it just after. But it's hard to answer this question. We know that at the beginning of our lineage, Paranthropus, Australopithecus, they were having more plant-based diet. I say we know, but that's what we suppose. And at more and more, hominins ate meat over the course of human evolution. And at some point, they started to exploit marine resources. So the question is who, when, where? And some researchers believe that it's something that only modern humans did. But other researchers showed that other hominins, such as Neanderthals, were eating marine resources. And this is evidences that you cannot discuss. However, yeah, and I could just say as well that for Homo erectus, it has been shown also in one side that they were associated to freshwater fish or turtle consumption. So no, it's not the coast, but it's still like aquatic food consumption. But what has been argued is that maybe this consumption was not a frequent consumption, which doesn't have the same impact on evolution. And so to answer this question of is it frequent or not, what's the proportion of different food categories in the diet? The best tool, from my opinion, the best tool is isotopes. And that's why I want to talk about isotopes today. So what is an isotope? So sometimes when I explain it, it's a bit scary because there's protons, neutrons. So I'm going to try to explain it in a more simple way. So you know many names of elements like carbon, nitrogen, oxygen, sulfur, copper, iron, radium. All those names are names of elements. And those elements are made of atoms. And those atoms, you have three examples here on this slide for the carbon. Those atoms can be heavy or light. You have here, for example, the carbon-12, and the carbon-12 is lighter than this carbon-13, just because it has one neutron more in the nuclei. But there are some, and here you have, for example, the example of the carbon-14, which is even heavier. But today I want to talk more about those atoms. Those different atoms are actually isotopes of the same element. So carbon-14 is a radioactive isotope because it decays over time. And those two guys are stable isotopes because they're stable, they do not decay over time. And when we do isotope studies, we look at the proportion of heavy, such as the carbon-13, and light, as the carbon-12, isotopes in any material. So I'm going to try to explain it also, what we do exactly in a very simple way. So I try to, like, let's take a bone and let's try to picture it as a ball pit. So the ball pit is full of balls, of course. And let's imagine that the blue ones are the light isotopes of carbon, and the red one are the heavy isotopes. And so what we try to do is not to quantify how many balls of carbon is in this ball pit, but more what's the proportion of the blue ones and the red ones. So it can be full of heavy carbon isotopes or full of light carbon isotopes. And so you can do this for carbon isotopes, but you can do it for many elements of the periodic table. And now we have more and more technology, and we can explore like almost all the elements and try to see what kind of information they can bring us. And what kind of information they can bring us, I will just talk about it a bit later. But just maybe I should say that, of course, some elements are difficult to study because you don't have enough of this element in a bone or in a tooth. You need a sufficient amount, and you need also to have at least 2 stable isotopes, because we look at the proportion of heavy and light. But that's basically it. That's not really important, but yeah, I just wanted to tell you we cannot do any element, but we can do a lot. And so now the question is, what can we analyze when we do archaeology? You might know that usually we only have 2 types of material left, the teeth and the bones. And maybe you know that the bones are made of a mineral, that's the bioapatite, and organic matter, the collagen, the protein. And so the collagen is made of carbon, nitrogen, and sulfur. And so you can study, well, it's made of other elements, but basically it contains a lot of carbon and nitrogen. And that's why when we study carbon and nitrogen isotopes, we usually analyze the collagen. And when you want to study other, like isotopes of other elements, you can work on the mineral parts of the bone or the teeth. And it's interesting because usually it's preserved for a longer time. So that was maybe a long introduction about isotopes, but I just wanted to give you this overview so that maybe it's easier then to follow why we do some studies in some areas and not everywhere, and what we choose some isotopes and not all the isotopes. But let's see now what those isotopes can teach us about aquatic food consumption. And for this, I'm first going to talk about carbon and nitrogen through 2 examples that have been very important for like studies in prehistorical times. And so one is at the onset of the Neolithic, and one compares Paleolithic modern humans to Neanderthals on a dietary point of view. So for this, I need to explain how carbon and nitrogen works. So how they work. You have here this example where I tried to make the plot as simple as I could. And so you see here this delta notation, it just express the proportion of heavy and light isotopes. So if you have higher values, you basically have more heavy isotope in the animal that you're analyzing. So basically, you see for carbon isotopes, usually marine animals, have higher values than terrestrial animals. And so if you eat fish, then your body will be enriched in these heavy carbon isotopes. So you will look like a fish from a carbon isotope point of view because you are what you eat. And so same if you eat like terrestrial food, you will be more in this area of the blood. And for nitrogen, it doesn't work the same way. So basically, depending on your position in the food web, you will have low or high values. Basically, a carnivore will have higher value than a herbivore. So every time you change what we call the trophic level, and the values become higher and higher. And so this principle has been used now almost 20 years ago in a Nature paper. So it was a major discovery where Richards and collaborators, they look at the carbon isotope composition of different humans that were coming from the Atlantic coast in Europe. And so they were from different ages. You see the age on this axis. So here they are the oldest individual and here the youngest. And you have the carbon here with the lower value, the lowest values here and the highest values here. So here it's more in the direction of fish consumption and here terrestrial food. And so what you might see immediately is that before this period, which was the onset of the Neolithic, so when you had more like fishers, hunter-gatherer, you have the carbon that kind of vary, and this means that people were eating fish, not always the same amount, but this adaptation to the coast was existing. And suddenly, when you have the beginning of the agriculture, like the Neolithic is when you have this beginning, people stop eating fish. Like it looks like they like it on a isotope point of view. So it's a quite interesting results because if you look at archaeological evidences, you will see sometimes for Neolithic times evidences of marine food consumption, like fishing. And those isotopes are telling us, well, it looks like it stopped being a frequent thing to eat fish at the onset of the Neolithic, which can be surprising, but it really looks like this. It has been confirmed in many areas in the world. So that's really interesting for the question we were asking, like isotopes, they can trace if marine food consumption was frequent or not. So let's look at the Neanderthals. You know, I was talking about the Neanderthals that were exploiting marine resources. Is it something frequent or not? Let's analyze Neanderthals. However, this is not easy as we would like because those analyses, I said, we make them in the organic matter of the bones, which is not always preserved. And also the Paleolithic sites in Europe, because of the change of the sea level, they're not so often preserved. So it's two difficulties that trigger the fact that we do not have any data on coastal Neanderthals. But we can try to see what's going on inland and not marine food consumption, but freshwater fish consumption. And so on this simple plot that I showed you before, I just added a fish just to show you that fish usually have quite high value for the nitrogen isotopes. And for carbon, they kind of vary around what we see in terrestrial food webs. And so many researchers analyze some Neanderthals and modern humans. And what has been seen when you compile all the values is that usually Neanderthals have values that are around or slightly higher values than the carnivores from the same sites. So it has been interpreted as they were super carnivores. And modern humans, they tend to have values that are higher for nitrogen isotopes And for carbon, it's usually a bit lower, but it could fit with fish, freshwater fish consumption, because basically, like I told you, every time you eat something in nitrogen, you go one step above on this plot. And so that would fit with modern humans eating fish that are around those values. So that has been for years the traditional interpretation of those carbon and nitrogen isotope values. But if I say that, it means that it gets more complicated. So indeed, 2 years ago, we published a result for Neanderthals that was plotting with the modern humans. And so this was really exciting, because at first when we saw this value, we were like, wow, we have the first Neanderthal with an evidence of a frequent fish consumption, which has many implications to understand human evolution. But we thought, okay, maybe there could be also another reason why this nitrogen isotope value was high. And also, like 2 years ago, there was like a interesting study that I'm going to detail after, that showed modern humans with super, super high nitrogen isotope values, and it started to be difficult to explain it just with freshwater fish consumption. So basically, we were reaching the limit of this method, and researchers now try to add up another one that's similar, but that allows us to go more into detail. Sorry. And so, excuse me, this is what I'm going to present you now. So I'm going to try to explain you this method, which you're going to see is not so complicated, because it's the same idea. We analyze nitrogen isotopes, but instead of working on the collagen that you see here, we work on the amino acids that are actually making the fibers of the collagen. So we take those amino acids, those molecules, separately, and we analyze separately the nitrogen isotope in them. So why is it interesting? Basically, if you take the herbivores, the nitrogen isotopes of two amino acids, the glutamine and the phenylalanine, will vary the same way. I mean that the higher the value is for one amino acid, the higher value it is for the other one. But if you change the position on the food web, if you compare herbivore to carnivore, it will change the value for the nitrogen isotope ratios for the glutamine. It will be higher, but not for the phenylalanine. So basically, all the herbivores will be on one line, and all the carnivores will be on one line on this plot. And same for the fishes. So basically, if you want to know what an animal or a hominin ate, you just need to plot it here, and you know what they ate. So it's kind of easy and fabulous. And so it has been done for Neanderthals. So you see here an example that has been published by NATO and collaborators, and they studied some Neanderthals that were showing traditional nitrogen isotope values in the collagen as a bulk. And you can see these results for those two guys. And you can see that they're just below this line, which is the carnivore line. And the herbivore line is here. So basically, it means that they were carnivore or omnivore with a lot of meat in their diet. So it didn't really change our interpretation of view on Neanderthal diets. And here, this is our study where we had this Neanderthal with a value that would be traditionally interpreted as freshwater fish consumption. And this Neanderthal was on the carnivore line. So there's another explanation why the nitrogen isotope ratio was high, but it's not freshwater fish. But then you might ask, all those modern humans that were having the same values, were they really eating fish? And that's a very interesting question that Drucker and collaborators answered with those modern humans from Burankaya, so in the Crimean Peninsula. And they were the ones with super, super high values for nitrogen that I showed you before. And so they did exactly the same study, and you have here the results, and you can see the herbivores on the herbivore line, normal, the wolf on the carnivore line, perfectly normal. In here, you have some foxes that can be omnivores, and the humans with super high values that would be interpreted as a lot of fish consumption. They are actually on this plot simply omnivores. And the fact that their nitrogen isotope ratios are high is probably just like you need to see they're just above this value for the phenylalanine. And this is mammoth. And mammoths usually have high nitrogen isotope ratios. And so it's possible that humans and also sometimes Neanderthals were showing high values of nitrogen, not because they were eating fish, but because they were eating mammoths. So that's what I wanted to say about those two examples. So as a summary, no fish consumption in the Neolithic, almost no, not a frequent one at least. And this fact that we first thought that what distinguished modern humans from Neanderthals were the fact that humans were fishing, but we really need maybe to do more of this new type of analysis to be sure that is really happening, because it doesn't seem to be always the case as you just saw. And so I would be interested to know what Erich Fischer thinks about this. But so we will see this later maybe. And I'm just going to move to the second part of this talk, because you see that I discussed about, okay, what isotopes can bring, but not the question of when did this consumption started to be frequent. So for this, I want to talk about how can non-traditional isotopes help to identify frequent aquatic food consumption. So again, we just saw that we could document a bit the fish consumption of Neanderthals and Homo sapiens. So now there was this question of when did it become to be frequent? Like, why wouldn't we look at Homo erectus, early Homo, and Australopithec, for example? Actually, those analyses I said were done on the collagen. And this organic matter is not preserved beyond 50,000 years in general in Europe, and even less in tropical and arid food webs. So basically, all the, let's say, key evolutionary steps in Africa or Asia, you cannot study them with those isotopes. But it's good because the technology is changing over time, and now we can work not on the collagen, but on the mineral of not the bones, but the teeth. Not the bones just because when the organic matter disappears, the bone becomes porous, and so you might analyze the soil instead of the bone. But when you work on dental enamel, then you can have a signature preserved for millions of years. So we could study many elements that are contained in the teeth, but my favorite one is zinc. And so I'm a bit biased there, but also I'm going to show you that we start to have evidences that zinc could be really interesting to trace the origins of coastal adaptation. And this is what I'm going to show you now. So the first evidence that zinc isotopes were related to the diet was shown by one team actually in Belgium, where they study vegetarians and omnivores, like they study the zinc in their blood, And you can see that the values are higher for the vegetarians compared to the omnivores. And then we had, and they analyzed different food products or plants, and people realized that the plant product tended to have higher values than animal product. There's some overlap, but in general, that might explain why vegetarians that eat plants have higher values than omnivores that do not eat plants. Sorry, that do eat plants, but that do eat animal products. So then this, you might have this difference. And so we first wanted to see if it could be recorded in bones and teeth. And we analyzed modern food webs from 3 sites in Africa: Western Cape, Kruger Park, and Kubifora. The 2 first sites, Kruger Park and Western Cape, it was on a broad geographical area. And we saw that carnivores tended to have lower values than herbivores, but there was a big overlap. But when we work on a very small geographical area in Kubifora, and that we removed the mammals, the non-mammal species, we could see very nice differences between herbivores and carnivores. So that was pretty encouraging to use it as a tracer, as a dietary tracer. So first in terrestrial food webs. But we need to check now if it was preserved for very ancient times. And that's what Nicolas Bourgon, so a PhD student that's supervised by Thomas Tutken and myself, that's what he did at the Max Planck Institute for Evolutionary Anthropology, working on a Pleistocene food web in Laos. And for this food web, we had a lot of carnivores, a lot of omnivores, a lot of herbivores. And basically, we tried to extract the collagen from those animals, and we were not successful. We had it only for 40s, and it was badly preserved. So basically, you had no collagen preservation, and we wanted to see, can we say something with the zinc? And you have here the results. So you can see the carnivores that have the lowest values compared to the herbivores that have the highest values, and the omnivores in between. And this is exactly what we had in Kubifora, except we didn't even have omnivores there. So here it's really cool. You really see that it seems to work. And also when you look at primates, like orangutans, they're almost herbivores. And they do overlap with the herbivores on this plot. Whereas the macaques from those sites, it seems that they were eating more animal products and they have lower values than the orangutans. So it would mean that this kind of variation of diet can be traced among primates. So again, really exciting results. We also studied Arctic marine mammals. And you have on this plot different results for zinc. So again, the lowest zinc is, the highest the trophic level is supposed to be. And for nitrogen is the opposite. The higher the values is, the higher the trophic level. So by trophic level, I mean position in the food web. The higher the position in the food web is. And we had some polar bears that eat seals. And you can see that the values for the polar bears are lower for zinc and higher for nitrogen. So it seems to really work also on marine animal with elevated position in the food webs. And you tend to have a correlation between those 2 traces. And here you can see that was for Kubifora, the terrestrial food web. The zinc works even better to differentiate carnivores from herbivores, terrestrial ones, compared to the nitrogen for this very specific site. So very exciting result because it works for terrestrial food webs, works for marine food webs. So very promising. And what's also very promising is something that has been shown by Jeremy McCormack, also at the Max Planck Institute for Evolutionary Anthropology. So for this study, it was like the second part of the one I just showed you, but we wanted to know how the zinc isotope will be impacted by the fact that some seals or polar bears were coming from some different geographical areas in Arctic Canada. So many sites were sampled for zinc, but also carbon and nitrogen. And so you see all the names, all the codes for those sites here. And you see here the carbon isotopes, nitrogen isotopes, and zinc isotopes for seals and polar bears, seals in brown, polar bears in pink. And what you can see is, let's take nitrogen isotopes. Depending on the site, you can have some variation, whereas they're supposed to eat kind of the same thing. And for zinc, you have some variation, but less variation. So that would mean that zinc is maybe less impacted by environmental factors for its ratios than nitrogen, which is the classical traces. So classical tracer. So I think, of course, I'm I'm promoting the zinc because this is the focus of my work. But this is why I'm focusing on this element, because I think it's a really exciting one and really promising one. And so I just would like maybe to finish on the zinc isotopes. I'm going to give some limits just after, but another illustration that is promising has been provided by another study that was not made initially in that direction, but we were analyzing some teeth of inland populations in France, and you can see their values in yellow here, around 0.7 per mil. And we analyzed also modern individuals from France that were having much lower value. And so the question was, are the modern individuals, like people born in the 20th century, because those guys, sorry, I forgot to say it, they were from the 17th, 18th, 19th century. So if the 20th century individual have lower values, is it because now the environment is polluted in light zinc, so zinc with low values? Or is it the fact that nowadays, even if you live inland, you can buy a kind of tuna, you can buy salmon at the supermarket and put it in your refrigerator or your freezer. And so to answer this question, we needed to analyze some humans from historical periods, so pre-industrial periods, but eating a lot of meat and fish. And we had the perfect candidate in Brittany with a population made of nobles and friars. And here is where they plot. So right next to the modern individuals in terms of zinc as the values, okay, they're a bit higher, but they're overlapping. And so they're definitely lower than the rest of the historical inland population we studied. So that was going in this direction, that with zinc, we could trace the fish and or meat consumption in human diet. But I'd say I would try to bring some nuances and maybe say that the zinc is not as fabulous as what we would like. And so I just made briefly, I'm sorry, it's a bit ugly, but this last plot where I decided to put all the data that I had for all the sites that we analyzed and that were published or about to be published. And you can see that there's a lot of overlap between the different colors. So those are actually families of animals that are usually mostly herbivores. Here you have some carnivores, like the Carnivora family, which most of the time has a carnivore diet, and that explains the names, but not for all of them. And you have so like those terrestrial carnivore and marine carnivores. And you can see here the primates, and in this case it's mostly the humans, but also macaques and the orangutans I show you. And this is why I wanted to say, okay, let's be careful with zinc still, because you can see that the primates tend to have lower value than carnivores. Whereas all those primates, for most of them, were either herbivores or omnivores, and not carnivores. So that would mean that maybe in addition to the diet, there's something in the zinc that's more related to the metabolism, because the metabolism among primates may be more similar than the metabolism between a human and a lion. So that might explain some nuances. And this is what we are focusing on now in a group around the ERC project, so the ICArEHB group. And so we're focusing not on this variation among families, but we are really trying to see how zinc isotopes could track the beginning of coastal adaptations in human evolution. And for this, we look at, this is the work of Jessica Mendes Cardozo, how zinc isotope can trace mollusks and fish consumption. Benjamin Ferrer, who's a postdoc, worked on how you can differentiate fish from marine mammal consumption. Pauline Méjean is doing a database of all the food products that she can, so that we can see how it varies between sites. Mathieu Leisen is developing a new method that allowing to do analysis that are not destructive, and therefore you could analyze some very precious ancient fossils. And myself, I work with all of them, but also I'm looking at an example that I just showed you before, the onset of the Neolithic, but specifically in Brittany, where maybe we had more fish consumption than what was previously thought. So as a conclusion, I'm afraid my talk might have been frustrating for you, because I didn't answer when and where were the origins of coastal adaptation, because basically until now we didn't have the tools to really understand when this marine food consumption became frequent. But I hope that if I come back in 3 years and be in front of you, I will be at least able to bring you some elements of the answer. But I'm sure that Erich is going to give you some insight that are not provided by the isotopes, but more on archaeological point of view with other tools, and that will be of very high interest. I'm really looking forward to listening to his talk. But before that, I would like to thank you for your attention. Thank you.
Hello, everyone. It's great to talk to you today, and thank you all for attending this ICArEHB Dialogues seminar about the origins of coastal foraging. Dr. Jaouwen's talk has provided some interesting and exciting information about the role of isotopes, not only to track diet, but also disentangle terrestrial and marine dietary contributions, which I think is pretty awesome. So here, I'm going to pick up on many of the themes that Dr. Jaouen mentioned and put them into the broader context of hunter-gatherer adaptations, focusing largely on studies from South Africa, where there's been extensive prior research. I'll then provide a brief summary of our current understanding about the prehistoric use of aquatic resources, both freshwater and marine, and various ideas about when and how these behaviors may have evolved. Then I'm going to briefly introduce you to the P5 project in South Africa, where my team has recently published on the first direct evidence of coastal foraging during a glacial maximum in southern Africa. I'll summarize some of our latest findings and also show you how we're studying the mobility and diet of coastal hunter-gatherers during glacial periods and across glacial interglacial phases. In many ways, the story of human evolution during the Pleistocene is about the preferential survival of individuals with increasingly bigger brains relative to body size, and the search for the dietary energy protein, vitamins, and essential nutrients to meet the increasingly greater metabolic energy demands of those big brains. Today, around 60% dry weight of the modern human brain is highly enriched in long-chain polyunsaturated fatty acids, especially docosahexaenoic acid and arachidonic acid. Studies have demonstrated that long-chain polyunsaturated fatty acids, iodine, and other nutrients are critical for gestation, neonatal and juvenile cognitive development, and they provide key health benefits to adults. The problem is that these fatty acids are essential, meaning that their primary source is dietary. Terrestrial game is a poor source of long-chain polyunsaturated fatty acids, particularly docosahexaenoic acid, because it's often absent from terrestrial food chains. This chart, for example, shows a sample of different food sources in Africa and from around the world ranked by DHA. What is most important to note here is that regardless of location, the freshwater and marine sources of fish and shellfish all provide significantly greater amounts of preformed long-chain polyunsaturated fatty acids. To put this another way, a hunter-gatherer is able to ingest 2 times more docosahexaenoic acid if they were to eat about a dozen mollusks versus the same quantity of warthog meat. But there's other factors that must also be considered. For example, DHA is not highly concentrated in terrestrial adipose tissue, and the fat content of wild game is also often very low. So the highest concentration of long-chain polyunsaturated fatty acids in terrestrial game is found in the brain, liver, and other organ tissues. These organs also spoil quickly, which makes it difficult to get them back to a camp if the hunters are far away. And they also make up about 15 to 30% of an animal's weight. Ethnographic observations have shown that it's not uncommon that hunters gut their kills, eat up some of the edible organs, and then dump the inedible, like the intestines. And there's also circumstantial ethnographic evidence that organ tissues do not always make it back to the demographic that would need them most, pregnant women and children. Among the Hadza, for example, even if organ tissues got back to camp, then there is still often male preferential access to those organs. However, among the Aceh, brain tissue is often reserved for children and the elderly. Terrestrial animals are also mobile, distributed widely and unevenly across landscapes, and they can be dangerous to hunt, meaning that there is generally low probability for successful hunts in these environments. Recent modeling results by Colin Wren and colleagues on South Africa's Cape South coast, for example, clearly demonstrate the influence of unpredictable terrestrial game hunting. Their modeling results predicted that terrestrial mammal resources accounted for only 15% of their modeled hunter-gatherer dietary intake, and this level was achieved only after simulating 12-hour foraging days for a population density that was below 20 people per 100 square kilometers. In contrast, shellfish from coastal environments are much less dangerous to exploit compared to large terrestrial game. They also require no specialized equipment to collect, and in fact, many modern-day coastal foragers simply use their hands to pry clumps of shellfish off of rocks. But most importantly, shellfish are largely immobile, making them very predictable to find. What this means for a hunter-gatherer is that if they're able to make their way to the coast, then they're pretty much guaranteed something to eat there, especially when you consider that there's a lot of other resources there too, such as shorebirds and eggs, beached whales and seals, and potentially also even fish. In fact, recent marine ethnographic studies of modern South African coastal foragers demonstrate that under optimal conditions, they can achieve energetic returns up to around 3,400 kilocalories per hour for men and around 1,900 kilocalories per hour for women. And that often exceeds the returns from terrestrial game. The caveat is that monthly tidal cycles restrict the greatest intertidal foraging potential to only about 2 to 3 hours per day for about 10 days per month. And intertidal return rates are otherwise lower depending on weather and water conditions. Interestingly, Wren et al. Also calculated that plant foods accounted for up to 75% of their model daily hunter-gatherer energy intake, which roughly accords with the 70 to 30 plant-animal food ratios for prehistoric hunter-gatherers suggested by Connor and Eaton and others. The high contribution of plant foods in hunter-gatherer diets can be explained partly by their foraging return rates. Another recent study by Bota of plant foraging return rates in the same study area, for example, showed that while plants are unevenly distributed across that landscape, they're generally aseasonal, and importantly, there are hotspots where edible carbohydrate-rich resources, like geophytes, can contribute up to 2,000 kilocalories per hour. Plant foods are also a resource that would have been accessible to men, women, and children. Carbohydrate-rich plants like tubers are especially important for the healthy function of the brain, red blood cells, kidney, and reproductive organs. Carbohydrates also offset nitrogen toxicity that results from eating too much lean meat, like much terrestrial wild game. However, plant foods like terrestrial game meat overall contain little or no long-chain polyunsaturated fatty acids. The fermentation of carbohydrates in the human upper gut results in the production of short-chain polyunsaturated fatty acids like alpha-linolenic acid. Humans can synthesize these short-chain fatty acids into long-chain fatty acids, but it's rather inefficient. So, where does this leave us? The evolution of the human brain required increasingly greater amounts of long-chain polyunsaturated fatty acids, among other nutrients. Terrestrial plants and animal foods do provide some of these nutrients, but not in high amounts, and these resources themselves are also often patchy and unpredictable. In contrast, coastlines would have provided a centralized location between land and sea, and their diverse terrestrial and marine plant and animal foods that, in many cases, were also both abundant and predictable, offsetting the risks inherent to each resource. Coastlines are also an environment that is naturally buffered against major climatic and environmental changes, providing a consistency of resources to hunter-gatherers over long time periods. My point is not that terrestrial environments were bad, because there is certainly abundant archaeological evidence worldwide of people living successfully in these kinds of environments. Rather, when you compare coastlines to terrestrial inland environments, the key difference isn't between the presence of any single resource in those places. It's about the mosaic of resources that each of these places would have provided to people, and coastlines simply offered more and in an environment that was less prone to severe climatic fluctuations. The earliest current evidence for coastal foraging has been found in small, isolated remnants of deposits adhering to the walls of cave PP13B at Pinnacle Point, South Africa. The marine fauna within those deposits indicates systematic exploitation of brown mussel from upper intertidal habitats as early as 167,000 years ago. The narrow selection of brown mussel and systematic collection of those mussel around tidal changes, though, suggests that while the Pinnacle Point records are early, they aren't necessarily the earliest. So, how far back could coastal foraging have gone? Unfortunately, there's really no good way to answer that right now, but there are some intriguing ideas. For example, John Parkington, Michael Crawford, Lee Broadhurst, Stephen Kunani, and numerous others have hypothesized about the influence of aquatic and especially intertidal foods on modern human cognition. According to these scholars, an influx of nutrient-rich marine protein drove the encephalization and cognitive evolution of anatomically modern humans after 300,000 years ago, leading to behavioral changes and novel innovations characteristic of the modern behavioral repertoire. By comparison, Curtis Marion has hypothesized that coastal adaptations followed the development of advanced cognition. According to his idea, the evolution of modern human working memory and executive functions during glacial marine isotope stage 6, which is during the Middle Pleistocene, enabled foragers to link complex phenomena like tidal and lunar cycles, allowing them to access deeper tidal areas safely and to schedule coastal foraging trips to balance between terrestrial and coastal resources. Thus, according to this idea, advanced cognition anticipated coastal adaptations. The problem is that the PP13b records, much like all other early coastal foraging records themselves, really aren't a good bellwether to predict how ancient coastal foraging may be. The reason is because these records provide only a very brief glimpse of early coastal foraging, and one that was dependent largely upon climatically driven sea levels that influenced the movements of coastlines and their proximity to the archaeological sites that we study today. So, if we were to speculate, how ancient could coastal foraging be? On the one hand, we can look to the antiquity of aquatic resource exploitation elsewhere in Africa. In the Turkana Basin of Kenya, for example, abundant freshwater catfish and turtle remains indicate hominins there have been exploiting aquatic resources opportunistically for nearly 2 million years. On the other hand, we can also look at non-human primate exploitation of aquatic resources. Numerous non-human primates are known to forage for a variety of aquatic and intertidal resources. In the case of the Burmese long-tailed macaques, they even make different types of stone tools to collect and process different intertidal prey. So, would an advanced cognition truly have been necessary for early and likely mostly incidental coastal foraging? Another intriguing insight into the origins of coastal foraging may be also found in the genetics of plant food adaptations. The earliest definitive evidence of cooked starches dates to 120,000 years ago at the coastal site of Colossus River in South Africa and about 170,000 years ago at Border Cave, also in South Africa. But there is genetic evidence that implies much earlier starch consumption. Modern humans have a high number of duplications in salivary amylase genes. These genes encode for the enzyme that breaks down starch in saliva. The selective suite of amylase gene variability is estimated by Inchley et al. To have occurred after the modern human Neanderthal split, sometime between 370,000 and 600,000 years ago. The high diversity of amylase genes is hypothesized to represent evidence of early adaptations to starchy diets. Additionally, a derived genetic adaptation in the FADS gene cluster is similarly estimated to have occurred between 200,000 and 500,000 years ago, placing the evolution of that gene cluster within the context of the evolution of the Homo sapiens lineage, around 300,000 years ago. The FADS gene cluster influences the human ability to synthesize short-chain fatty acid, alpha-linolenic acid, into long-chain fatty acid, docosahexaenoic acid, and that derived allele improved that efficiency by about 4%. Scholars have speculated that this derived FADS gene was an adaptation to short-chain polyunsaturated fatty acids common in plant foods, as people moved away from primary sources of long-chain polyunsaturated fatty acids found in aquatic environments like coastlines. Yet, something that I've been thinking about lately is, what if this was actually a coastal adaptation? Early Homo sapiens populations were highly mobile, so it's unlikely that any groups remained along coastlines for long periods of time, rather than intersect them opportunistically during their annual movements around the landscape. A coastal population of early Homo sapiens, therefore, would have had a regular but inconsistent supply of docosahexaenoic acid and arachidonic acid in their diets from intertidal foods, but perhaps also seabirds, scavenged marine mammals, and eggs. The rest of their diet would have been a combination of lean meat from terrestrial animals, but also carbohydrate-rich plant foods. If the amylase and FADS genes evolved around the same time, prior to or around 300,000 years ago when Homo sapiens evolved, then is it possible that a high contribution of carbohydrate-rich plant foods could have been particularly advantageous to early coastal populations, not terrestrial populations? It comes back to the idea of environmental mosaics that I mentioned earlier. And what I've been thinking about is that while a carbohydrate-rich diet would have provided energy and other nutrients, having an increased ability to synthesize short-chain polyunsaturated fatty acids into those long-chain polyunsaturated fatty acids, even minor, would have ensured a more consistent and consistently high supply of long-chain polyunsaturated fatty acids in the diets of early Homo sapiens populations living in coastal contexts, and that would not have been available in coeval groups living in more inland locations. But that's perhaps speculating a little too broadly for this talk, and it's something best to consider later. What I want to do now is return to one of the major underlying problems in the study of the origins of coastal foraging that I mentioned earlier, the lack of long-term and continuous records of coastal foraging history. During the last 200,000 years, the Earth has undergone repeated changes in climates. During glacial phases, more oceanic water is trapped in polar ice caps, leading to drops in global sea levels, whereas during interglacial phases, the ice caps melt, resulting in higher global sea levels. In southern Africa, where the earliest known evidence of coastal foraging can be found, there is a mostly very broad and very flat continental shelf, meaning that drops in sea levels there during glacial periods translated into major changes of the shoreline, ultimately exposing as much as 80,000 square kilometers of seafloor during glacial periods, which is approximately the size of the island of Ireland today. That exposed landmass is believed to have provided a significant range expansion for grassland habitats and grazing taxa on places like the Paleogallus Plain, making it attractive to hunter-gatherers. However, when important early coastal sites like Pinnacle Point, Blumbus, Claces, and so many others became inland sites during glacial times, hunter-gatherers seeking coastal resources had to move out furthest to remain within daily foraging range of the coast. The large gaps we now find in coastal foraging records are, therefore, due largely to rising sea levels during interglacials, sweeping across the exposed continental shelf and obliterating traces of glacial human occupations in those areas, leaving us today with only short and disconnected interglacial excerpts from a much larger story about people's relationship with the sea. In fact, based on my calculations, up to 50% of the last 300,000 years of potential coastal foraging history may be lost to rising sea levels. Mpondoland on South Africa's eastern seaboard is important to the story because it offers a rare opportunity to fill in those gaps. But how? The reason can be found just offshore. Unlike the Agulhas and Atlantic continental margins that are broad and flat, Mpondoland has a very narrow continental shelf that capped coastline movements during glacial periods to just 8 kilometers from the modern coastline. The ship seen in this image, for example, was observed from our coastal site at Waterfall Bluff. The ship was following the continental shelf break, and it was clearly visible to the naked eye. This means that past hunter-gatherers living on today's coastline would have always had access to coastal resources during both glacial and interglacial phases, and the archaeological records in Mpondoland are less likely to have been destroyed by changes in sea level. I developed the P5 project as an outgrowth of my work with Curtis Marion and his team working at Pinnacle Point. Our initial survey in Mpondoland in 2011 documented nearly 24 rock shelters and open-air sites ranging from relatively recent European colonial shipwrecks back to the Middle Pleistocene, early Stone Age. Yet back then I didn't think that my team and I were ready to tackle the complexity surrounding those Middle Pleistocene sites that we had found. Instead, I turned my attention to another site that we had documented, at Waterfall Bluff, where our early data suggested occupations dating to the late Pleistocene, perhaps even the last glacial maximum. The last glacial maximum. It's the period of maximum ice sheet expansion during the last ice age, dating from 26,500 to 19,000 years ago. Sea levels were nearly 130 meters lower than today, which is about 400 feet. leading to widespread exposures of continental shelves worldwide. In South Africa, the exposed continental shelf was greatest at the Paleogallus Plain, and it was also generally cooler than today, though not necessarily drier in all places. And during the last glacial maximum, records of coastal foraging across southern Africa cease. Evidence of coastal foraging does not reappear in abundance until about 13,000 years ago. And the disappearance and reappearance of coastal foraging in the archaeological records can be linked directly to changes in sea levels that pushed coastlines beyond the daily foraging distance for hunter-gatherers, which is about 10 kilometers, or roughly what a person can walk out and back in a day. Knowing this, I looked to Waterfall Bluff as a proof of concept to test a hypothesis. Places with narrow continental shelves like Impondoland have the potential to preserve long-term records of coastal occupation and foraging simply because those narrow shelves would have limited past coastline movements across glacial and interglacial periods. Before now, though, a coastal glacial maximum record did not exist. Our work at Waterfall Bluff changes that, and our recent publication in Quaternary Research documents, for the first time, repetitive coastal occupation during the last glacial maximum and the turnover from the Pleistocene to the Holocene. To achieve this result, we started a collaboration with South Africa's Itemba Labs in 2019 to create one of the most comprehensive radiocarbon chronologies for the late Pleistocene in southern Africa. This record was developed from 51 AMS radiocarbon ages. It accords with our optically stimulated luminescence chronology developed at the University of Wollongong, and combined, these records document repeated human occupation at the shelter from 38,000 years ago to around 11,000 years ago, spanning the last glacial maximum and the last glacial interglacial transition. We've relied on this chronology to date another important finding: our multi-proxy evidence of coastal foraging from deposits dated to marine isotope stage 3, the last glacial maximum, the last glacial interglacial transition, the early Holocene, and the middle Holocene. When many people think about coastal foraging, attention is often directed towards seafoods, and we certainly have those. At Waterfall Bluff, for example, the archaeological deposits are predominantly brown mussel with smaller quantities of limpet and even a few Natal rock oysters, all of which are common on rocky shorelines today. We also have evidence of marine fish and marine mammal exploitation. To date, we have identified over 1,600 pieces of marine fish bone, and the taxa are representative of species common today from estuarine environments. Most recently, we've even identified a juvenile Cape fur seal tooth from some of our last glacial maximum deposits. We've also discovered a variety of perforated marine shell beads, ostrich eggshell beads, as well as shaped bone tools. But I want to direct your attention to two worked bone pieces in particular. Rock art representations of fishing in the Eastern Cape and Free State show hunter-gatherers spearfishing and line fishing, sometimes from floats. At Waterfall Bluff, we've recovered a broken bifacial bone point, seen here center top, that is similar in size and shape to ones reported from Nelson Bay Cave. Additionally, we've also recovered a possible broken line fishing gorge, seen here at right. The size and shape of the worked bone object is similar to fishing gorges that have been found at Nelson Bay Cave and Alans Bay Cave. Microscopic analysis of these two pieces has been stopped on account of the pandemic, so further evidence that they were used for fishing activities remains circumstantial, but we do intend to follow up on this work as soon as possible. In the meantime, we do have evidence of another coastal foraging behavior that is rarely preserved: plants. Southern coastal forests are common in Impondoland. These habitats grow on coastal dunes and include numerous endemic species, including a large diversity of woody species. Our charcoal specialist, Dr. Allison House, has identified at least 3 different species of wood charcoal from our glacial MIS3 and last glacial maximum layers. The wood species she has identified are all common to coastal forests. In 2019, our marine ethnographer, Dr. Jan de Fank, observed that modern Namapondo women who come to the coast to collect shellfish also often took the time to collect wood for cooking fires during the same foraging trips. Not coincidentally, the preferred species of wood that they collected derive almost exclusively from coastal forests, and not from the nearby scarp forests or thickets. These records provide a window into hunter-gatherer behavior across a glacial-interglacial phase in a persistent coastal context, which has never before been studied in southern Africa. What these findings hint at is a diverse pattern of coastal exploitation at Waterfall Bluff. Not an exclusive focus on the sea, rather a centralized location between the land and the sea and their plant and animal resources that attracted people amid regional climatic and environmental variability. We've linked these data to our robust multi-proxy paleoenvironmental record, which, as you can see here, has been developed by a diverse team from around the world. These detailed records, published recently in Quaternary Science Reviews, show that all major vegetation zones were sustained during the late Pleistocene and the glacial maximum, including the fire-adapted grasslands and the water-loving scarp forests. This finding is corroborated by our offshore mapping showing incisions in paleo rivers on the submerged continental shelf that indicates freshwater runoff from precipitation during glacial periods. That precipitation not only would have supported local vegetation and terrestrial animal communities, but also people. But what about in the surrounding region? Archaeological records dating to the last glacial maximum, which are already rare in southern Africa, suggest widespread movements of people during this time. By 29,000 years ago, for example, sites in the western Lesotho lowlands are abandoned and not reoccupied until the very end of the last glacial maximum, around 20,000 years ago. The situation is similar in the eastern Lesotho highlands, where occupation persists until 23,000 years ago, but those sites too are abandoned until after the last glacial maximum, about 15,000 years ago. Lower elevations see similar population movements. In the eastern Cape Midlands, between the coast and the Mlooti-Drakensberg highlands, occupations also end by 25,000 years ago, and do not resume again until about 13,000 years ago. To the northeast, near the KwaZulu-Natal coast, the site of Umhlazana is abandoned by 30,000 years ago and only sees brief pulses of occupation at 14,000 years ago and around 10,000 years ago. Clearly, people are moving around, but where did they go? Our friends and colleagues working in the Mlooti-Drakensberg highlands have speculated that highland hunter-gatherers there were linked to the interior plateau, and particularly tethered to the Orange Senku River. Yet I think this hypothesis overlooks some important objects that were left behind by those highland hunter-gatherer populations. At the site of Sahonghong, for example, a vervet monkey scapula and marine tick shell beads have been discovered in deposits dating just before the site and the highlands themselves were abandoned 23,000 years ago. Vervet monkeys are a tropical lowland species, suggesting that either live vervet monkeys or pieces thereof were brought from the lowlands to the highlands, like the marine shell beads. Interestingly, when people reoccupy Sahonghong after the glacial maximum, they brought more shell beads from the Indian Ocean coast with them, plus more vervet monkeys, or pieces thereof. Meanwhile, hunter-gatherers at Waterfall Bluff had ostrich eggshell beads. Ostriches, though, are not endemic to the coast because it's too humid and the grasses too tall. The nearest likely contemporary location for ostriches at this time was probably the short grass plains of the interior Midlands to the west of Waterfall Bluff. This evidence raises two important questions about late Pleistocene hunter-gatherers. 1. Were the people moving, or just goods and ideas? And 2. Where are the people? It's possible some groups of hunter-gatherers did go to the interior, perhaps even along the Senku River, but even then, sites like Melekane at the headwaters of the Senku are still abandoned for thousands of years. What our work at Waterfall Bluff now provides is a new and clear signal that groups of hunter-gatherers occupied the coast throughout the last glacial interglacial transition. Our research has further shown that Afro-temperate forests, grasslands, and bushveld existed alongside freshwater rivers that created estuarine environments at the coast. And that coast stayed within 8 kilometers of waterfall bluff, even during the glacial maximum when people exploited it, as evidenced by marine fish and shellfish remains, as well as the charcoal of tree species from coastal forests. When we draw in the exotic material culture evidence from Waterfall Bluff and the highlands, those ostrich eggshell beads and vervet monkey bits specifically, it points towards movement between the interior and coast, and we're beginning to test these links via a multi-proxy study of prehistoric mobility patterns. First, we're getting ready to start a phylogeographic study of vervet monkeys. Vervet monkeys have distinct populations west and east of the Multi-Drakensberg Highlands. And to do this, we want to analyze their ancient DNA from the Sahong-Hung vervet monkey remains and see if we can link that ancient DNA to a modern population, perhaps on the coastal side of the highlands. Second, we've already made significant headway on a multi-proxy geostatistical isoscape model ranging from the coast to the Multi-Drakensberg Highlands. We're going to be able to use this model to test the movements of past people, animals, and goods like ostrich eggshell beads. Third, we're also planning to analyze the isotopic signature of our ostrich eggshell beads and map those isotopic signatures onto our isoscape maps and see where those ostrich eggs were collected. But while each of these data sets potentially builds a stronger case for links between highland and coastal lowland populations, none really directly tests the people themselves. So that's why we're also making preparations to potentially analyze our 2 LGM human teeth. Found in 2019, the lower second molar, which was unerupted, and a heavily worn lower incisor are among the most precisely described and contextualized human remains in Africa dating to the last glacial maximum. We've sampled every layer below, within, and above both teeth and 2 vertical transects, comprising 22 radiocarbon samples, which have been incorporated into that Bayesian chronology model I showed earlier. The teeth themselves are also mapped to millimetric accuracy alongside all other excavated materials within those layers. And we have multiple micromorphological samples currently under analysis that will give us granular level details about their stratigraphic contexts. Preliminary analysis of these teeth has already begun by Dr. Shara Bailey and one of her graduate students at New York University. In time, though, what we have planned is a comprehensive isotopic analysis of them, potentially even including zinc isotopes, as described by Dr. Jaouen. The isotopic study that we have planned for these teeth could potentially provide direct information about movements of prehistoric individuals during the formation of the teeth, perhaps even directly tracking movements between the coast and the highlands. And it could also provide important information about the diets of hunter-gatherers at that time, particularly seafood-rich diets. So, to wrap up our discussion, our latest work from Waterfall Bluff in Impondoland has documented coastal occupation during a glacial maximum and across a glacial-interglacial phase for the first time in southern Africa. These findings are significant, not only because they show a robust and diverse exploitation of coastal and terrestrial plants and animals during glacial periods, but because it also shows that the pre-existing gaps in coastal foraging records can be filled. And it opens up the possibility of one day building long-term and continuous records of coastal foraging history and to study the influence of coastal foods in human biobehavioral evolution across long time periods. I've really appreciated talking to you all today, and I hope you all have enjoyed it as well. Thank you all for listening, and I look forward to answering all of your questions.
Thank you so much to our speakers, Erich and Klervia. Thanks a lot. So now we are at this stage that we actually can start a dialogue of many voices. And to start that, and if you are not very shy, I would ask for as many of you as possible to turn on your cameras. So this way, Klervia and Erich can have a more personal feeling of their audience. So for the first question, in the chat, Gopesh, I hope I'm pronouncing your name correctly. Gopesh had a question for Klervia. Gopesh, do you want to go ahead and unmute yourself and ask the question?
Yeah, I'm so sorry if I have too much noise in the background. First of all, it was a mind-boggling session. Thanks, Eric, and thanks, Klervia, for such a wonderful session. The question I wanted to ask, zinc isotope has been quite wonderful, but regarding the nitrogen isotopes and sulfur isotopes, do you see any kind of diegetic changes in the values of delta N because of combustion or some kind of microbial degradation? And do you think by clumping the sulfur isotopic data, we can clearly or in a much higher resolution, we can demarcate between the terrestrial and the marine dietary sources?
Thank you very much, Kovesh. So I didn't understand the end of your question, but I think I read it in the chat. So to answer the first part of your question on the nitrogen isotopes, there were different studies on the bulk collagen, but not on the amino acids from what I know, showing sometimes that it has a small effect, sometimes not seeing anything. So it's unclear. And then it's usually not the way people interpret elevated nitrogen isotope values, but it has been argued, and there's some data going in that direction, that if your food is rotten or fermented, then it can trigger higher nitrogen isotope ratios. So that could be also maybe a reason why we see those values among hominins. What I would say is we don't know yet in terms of amino acid what would be the impact depending on different amino acids. And there's also many other possibilities, and maybe also a metabolic difference between hominins and other carnivores that are studied that has been argued. So there's many ways to interpret those elevated nitrogen isotope ratios. And to answer on I think you were asking about the sulfur isotopes. And the sulfur isotopes, they're a really good tracer of coastal adaptation, because with the sea spray, everything close to the coast has very high value for the sulfur isotope ratios. But this is true for terrestrial and like terrestrial food and marine food on the coast. So you cannot decipher the two. So if you have someone living on the coast, this person will have elevated values if this person is eating local food.
Just add up, do you also target any rare earth elements along with the zinc isotopes or the nitrogen isotopes? Any rare earth element based proxies to demarcate between the terrestrial and the marine resources, if I'm clear?
No, I couldn't hear. Can I see the difference between what and what?
Rare earth elements, any rare RE based isotopes?
Yeah, well, I didn't look into this, so I won't be able to answer your question, unfortunately.
Thank you. Any more questions to our speakers? I think Roz has a question. Feel free to unmute yourself.
Thank you both for your amazing talks. What my question was to Klervia was that why are Neanderthals always seen as top predators, and then humans who have higher nitrogen then just say, well, they're eating fish, so they're very intelligent. But yet the Neanderthals are seen as an animal, and so they are top predator, whereas the humans are like, well, you're very intelligent. So
yeah, maybe you have a point. I don't know if it was a bias like this at first. It's true, like when you see slightly higher nitrogen isotope ratios for the Neanderthals, you could argue that they were eating a bit of fish, just a little bit. And from what I understood, I think it was not the favorite explanation because I think no fish remains could be found on the sites where you observe those values, whereas fish were sometimes found on the human sites. And so and also like what you were usually seeing, it's much higher nitrogen isotope values for humans compared to Neanderthals. So for the humans, you couldn't say they're just super carnivore. You needed to find another explanation. And I think that's why you see this difference. But now we see Neanderthals with high values, and it is possible that there's an environmental component also in this that's playing a role.
Yeah, super interesting, Klervia. Thank you. Does the audience wants to chat, dialogue with any of your speakers? You can raise your virtual hand here in the emotions or just unmute yourself, actually. I think Godfra Deveve put something in the chat.
Yeah.
Godfra, you want to unmute yourself?
Yeah, sorry, I unmute. Yeah, no, but just for me, who's an ecologist, a terrestrial ecologist, I'm just thinking that coasts are very limited. And can you imagine that actually what you presented of marine evolution or marine coastal adaptation happens actually inland with the, is it like all the thing about the omega-3, is it a theory that also holds on freshwater bodies, and actually perhaps it happened around a lake or, I mean, you see what I mean, that, yeah, are the theories valid also for inland water?
Yeah, I mean, when you look at the papers, for example, the studies out of the Turkana Basin there, and they're showing early aquatic adaptations at about 1.95 million years ago, and that's cut marked catfish, it's Clarius species, as well as turtle carapace. I mean, you can kind of see similarities in the environments. Those lacustrine environments and riverine environments would have still provided numerous different kinds of resources. Fresh water, and that fresh water would have attracted different kinds of animals. And then you have the aquatic species to boot. And those aquatic species, depending on what they are, would have higher amounts of those brain-specific nutrients, those polyunsaturated fatty acids that are so essential for brain growth and cognitive development. So yes, when you look at the papers, for example, out of the Turkana Basin, they're arguing that, yes, it's possible that these hominins could have supported themselves in those kinds of environments. Still, though, I think that the diversity of resources is greater along coastlines. And certainly what coastlines provide, when you look at it in the much broader kind of a context, specifically like a global context, is that rivers and lakes are very isolated on landscape. So, you know, if you're along a lake or if you're along a river, well, you have this conduit. But if you're along a coastline, it's something that you can follow pretty much all over the world. And in fact, that's what humans did. You know, when they expanded out of Africa, that there's this idea, which I do believe in, that they followed coastlines. So I think that coastlines offered more, but certainly the idea that there was these aquatic adaptations and the nutrients and everything would have also been provided in inland aquatic environments as well.
Okay.
Thank you, Erich.
Thanks.
Darren, I see your virtual hand. Feel free to unmute yourself.
Virtual hand worked. Hi, thank you both for really interesting presentations, and I'm sure we could talk about this for hours. I was actually following up. I had an earlier question for Klervia, but then Ros mentioned the additional point about the fact that sort of you know, Neanderthals had high nitrogen values, but they're considered to be super carnivores, and when it happens to moderns and this kind of thing sometimes seems to, and you mentioned that it's because there's a lack of evidence of marine exploitation in the caves themselves. And my point to you is that being weighted with regard to what Erich was mentioning before, and a number of people, including myself, will be talking about, is the whole point of visibility of these resources given sea level changes. So which means that it's the same site, but the reality is you might be consuming marine resources, say in winter when you go down to the coast and you come, and this is an interior site so you're going to get high nitrogen values, but you're not going to find evidence of it. You're not going to bring that stuff back to the site. So I'm querying whether that is a good enough rationale to be able to debunk the one and not the other. You'd have to look and see the availability, the visibility of those resources. And the other question, which is also in your presentation, which because you mentioned on one slide that the zinc values were more consistent, I think it was in seals, and that was but they're incredibly far more variable in the polar bears, which were like the carnivores, which you didn't mention. But I wondered if you had something to say about that.
Yeah, so thank you for your question. So for the first question, I just need maybe maybe I wasn't clear with actually there were never nitrogen isotope analysis that were done on coastal Neanderthals, to my knowledge. So the sites I mentioned, they were inland sites. But of course, you definitely have a point that maybe there was like freshwater fish consumption and we're missing it just because it was not on site.
Or they're tracking resources, they're moving to
Yeah.
the coast and back in again or whatever.
Yeah, and this is also like, like there's more and more this idea among the isotope community that it's also related to mammoth consumption, and you don't always find the mammoth remains on the sites. And so is it like it was really eaten, or it was just not brought back to the site? So that's a good question, and that's I think we're reaching here the limits of what isotope can tell, and we need to use maybe more isotopes and also to combine with other archaeological tools. Sorry, what was your second question?
The second one was about the variability of zinc.
Yes.
You had a slide where it was quite, you pointed out that it was a very great consistency, more than say I think it was carbon, but
yeah, like
but it was very variable in the polar bear.
So one site in the polar bear, it doesn't make sense at all for the zinc. So the values are overlapping with the seals. And even though when you consider everything together, the trophic level spacing was more consistent for zinc than for nitrogen isotopes. So
Okay.
It's true when you look at it. I thought, okay, so it looks ugly, but when you do the statistics and you look at the numbers, actually, it works well. But I don't say like we need to do, of course, more studies and so on. But I was a bit surprised as well when I saw the figure at first, and when I saw the numbers to see that actually zinc was more consistent in terms of trophic spacing. But you have a point. Like there's one site we have no explanation for this, and the polar bear values make no sense.
Okay.
Vera, if I could follow up
Yes,
real
of
quick
course.
on the idea of the visibility with your question, Darren. Yes, I think actually one of the hardest problems in tracking any kind of coastal foraging behavior is visibility. Shellfish can be, depending on the environment, easily dissolved, and fish bones, for example, can be very tiny and overlooked. And I learned quite quickly with our fish specialist, Dr. Erich Zohar, that actually identifying fish bones is actually very, very tough. And if you don't know what to look for, then they're easy to overlook. So actually, when we were working at Waterfall Bluff, we did not originally see direct traces of coastal foraging, shellfish or anything else that was visible to the eye. We had seen a whole bunch of decalcified deposits, so we were kind of clued in that there might be something happening, but those earliest excavations were right along the drip line, and everything was actually being dissolved. And one of the people who was listening to this presentation earlier, she just left, Dr. Annette Ertel, and her PhD supervisor, Ket Saba, were the first people to actually find those evidence. They took sediment samples and they looked at them under a scanning electron microscope and were able to find microscopic traces of mottled shell. And we were able to link that back to mussel. That paper is actually almost ready, and we're going to be submitting that soon. And then from there, we kind of knew that what we had, and we started to work further into the rock shelter, and that's when we started to pick up the fish. And now we have preserved shellfish in better preserved deposits that are not away from that drip line. And we've also recently found a juvenile seal tooth as well from our last glacial maximum deposits, which actually is very recent. And I've been bombarding all of my colleagues with questions, and because that's really intriguing. So the idea about Neanderthals or even Homo sapiens in Africa, I mean, we could be missing a significant amount of these coastal foraging behaviors, particularly in open air sites. And that's something else to consider, because we're working in a rock shelter because it has increased preservation, but within walking distance of our work at Waterfall Bluff, we actually have sites that are along the coast that date back to 300,000 years ago. These sites contain early Stone Age Acheulean artifacts. They contain very early Middle Stone Age artifacts. And we've started working there. And now we're really starting to eye those sites more closely. But how are we going to recover if any coastal foraging evidence is preserved? It's going to be microscopic, it's going to be trace, and we're going to have to find proxies in order to actually recover that. I'm confident that we're going to figure that out, but it's going to be very, very complicated to do.
I'll just add to what Erich said, that he talks about microscopic evidence, so it's not something we catch right away. I'm biased, I'm a micromorphologist, I agree, but I would bump that up and say molecular nanoscale is where we are now starting to be able to go to answer some of these questions, with, for instance, lipid biomarkers or if preserved sediment DNA. So I think we will potentially scrape a bit more of this invisible record, which I think will be really neat. Anyone has a itch for a question for our presenters? That feel free to unmute yourself, or Curtis.
Yeah, hi everybody. I just wanted to say those were two fabulous talks. That was a great way to start the morning here in Arizona. I got to eat my breakfast while listening, and very impressive. I just wanted to say a few things about things that people probably already know, but not all coastlines are productive, right? So the intertidal zone varies wildly. Darren Fa has done great work on this, looking at the productivity of coastlines in the Atlantic versus the Mediterranean coastal areas. And so just to give you an idea, like on the South African coast, the West Coast has very high biomass, but very low diversity of intertidal shellfish. And then as you come into the South Coast and move to the East Coast, the biomass goes down and the diversity of species goes up. So up where Erich is, the biomass is lower, but the diversity of species is higher than it is on the West Coast. So the kind of intertidal strategies that hunter-gatherers use would vary across that gradient. But also, when you get up into East Africa, and I've spent a lot of time in East Africa back in the 80s and the 90s, particularly in Somalia, coastal Somalia and Kenya, those intertidal zones are not rich. And there really isn't a lot of evidence for intensive coastal exploitation of those coastlines until people start having sophisticated technologies like fishing lines and boats and so on. So while I've put a lot of emphasis on the importance of coastal adaptations in human evolution, I don't think you can follow coastlines as a hunter-gatherer out of Africa. The Arabian coastlines are very poor in the intertidal zone. You have to have really specific conditions. So I think the coastal adaptation, as people moved out of Africa and into Asia, Southeast Asia, kind of flickered in and out of significance as they intercepted coastlines that they could exploit easily. But then as they developed the complicated technologies, boat technologies and fishing technologies, then they could exploit the low productivity intertidal zones. But what I'm trying to get at here is that a really cool project that could be done, and it would require a big collaboration between lots of different research units, would be to map the productivity, the foraging productivity, right? The human foraging productivity of the African coastline, so that we could somehow have a continuous map up the West Coast, up the East Coast, you know, because when you get up to the Atlantic coast of Africa up in Morocco, it gets rich again because of, as Darren has shown. That would be a really cool project, and it would be really important to do. But like I said, you'd have to have a number of different research units collaborating to do that. And, you know, your new research unit at your university sponsoring this event would be a great one to kind of be involved in that. So I pitched that as an idea, as a big international collaborative thing to do that also would be a hell of a lot of fun.
I think our director, Nuno Bicho, is here somewhere, and I'm thinking very happy about that. See, he even shows up.
Yeah, that's a great idea, Curtis. I like it a lot.
Let's try to put it together.
Yes, for sure. Right.
Anyone else wants to follow up on the comments by Curtis or other questions to our speakers? If not, I'll take advantage that I'm the moderator, and I'm going to ask a slightly different question, which is something that always bothers me a bit, right? We always focus on, of course, in the origins. In archaeology, that's a lot of what we do, right? The first of. And we know that in terms of coastal adaptations, at some point in time in worldwide, you do have this intense reliance on marine resources. And we see it archaeological by the presence of shell middens, right? The most impressive one, like in Brazil, in the sambaqui that are 20 meters high, or in Australia, or in the Americas, etc. We always have that. And we have it, of course, here in Europe, namely during the Mesolithic. Some of the sites we study here in our center, in the Murge Mesolithic, they are impressive, monumental sized mounds, artificial, of shell middens. But they also collapses, right? So for instance, in the Mesolithic, the introduction of farmers, and whoop, you don't have it anymore. So I guess it's kind of the opposite of what's the title of this talk, which is the origins of. I would like to hear your guys' thoughts on the collapse of. Why does it collapse if it's so beneficial?
So I'm not able to answer why, of course, but it's a question that I find fascinating as well, and that's why I'm currently interested in what's going on in Brittany, because we have still a few shell middens from the Bronze Age, and so I also like I'm not an archaeologist. I'm more curious about the opinion of the people that I hear. But I always wonder if it's related to the fact that it was a culture that was coming from an area where there was not, before the Neolithic, a strong reliance on marine resources, and that it came like somehow as a modern culture and you adopt it. And like what we're trying to do now, I'm working with people doing ancient DNA analysis, is to see if those Bronze Age sites where you have still some reliance on marine resources, if they are the same population as the other guys that are also living on the coast but not eating those resources.
And as far as, for example, what we see in South Africa there in Mpondoland, the contemporary populations, they're Mpondo, and their ancestors moved into that area about 2,000 years ago, and a little bit earlier. And when that happens, you do see farming, herding, all that kind of activity coming into that place, but they're still going to those coastlines, and they're still exploiting those coastal resources. So it's not so much that there was at least a whole-scale shift away from from coastlines. It was more of an adaptation of integrating those food sources into their more terrestrial-based diets as well. And to this day, the local Amapondos, they might be living in towns. Some of these folks have to walk 2-3 hours to get down to the coast. And these women are doing those trips on a pretty regular basis, and it's predominantly women, and they're coming down to the coast and they're collecting not only those brown mussel, but I haven't mentioned, but we also have an endemic species of oyster down there, the Natal rock oyster that they're collecting, and then as I mentioned earlier, also different species of wood for firewood. So they're still utilizing those coastal resources. It's just the way that they've done that has adapted. It's changed.
Yeah, thank you. And yeah, I think it's a question that there's not an answer, also because probably it will vary, right? The same way that white starts will vary, white ends also will vary deeply from area to area and time to time. But I think it's something we often forget to think also.
Yeah. This idea of variability, if you don't mind, Vera, I want to kind of tie back into what Curtis had said, because I actually really like his idea about building this map, looking at the productivity of coastlines across Africa, and of course I've heard him talk about that for many years and everything. So, but one thing I certainly agree with him about the variability in the productivity and the biomass differences as you move across the coastlines of Africa. But something that and if you're looking specifically at those intertidal resources, then I think, yes, it would have a much greater impact. But then again, it comes back to this idea that if we were to do any kind of a project like that and develop this and build these maps, it shouldn't just be focused on, for example, intertidal resources. We also need to look at those nearshore terrestrial resources as well, because to me, the big benefit, the big draw of these coastlines is not just the marine foods. And I mean shellfish and fish, maybe a seal or a beached whale, seabirds, and even eggs and other things, but also those kinds of resources that would have been found on land right along, coast adjacent in those various different kinds of environments. And when you look at that, to me, really, as you move up, even as you move up the east or the west seaboards of Africa, those coastal environments still provide a greater diversity of resources in many different environments that you don't necessarily always get in terrestrial environments. So to me, what would be really exciting about these maps would be to take that slice from the marine, maybe inland, maybe say about 10 kilometers, and map that and look at that kind of a variability. Certainly in a place like Pondoland, what we see is that because of the presence of the Aldeias current right there, it's actually driving the climatic changes in that area that really don't extend very far inland. And so what we see along the coast, once you move in about 10 kilometers, it stops, and we don't really see those freshwater rivers or those other kinds of habitats for terrestrial plants and animals and everything else like that would have supported hunter-gatherers.
Very cool, thanks. Allison has a question in the chat about the topic that I know some people are also interested in ICAREHB. So Allison, if you want to unmute yourself, or I'll just ask your question. I guess I'll ask. Or is Allison here? No. But Allison was asking, is there any evidence for use or consumption of marine algae or seaweeds, especially on nutrient-rich West Coast?
I've been actually reading about the nutrient load of algae and seaweeds, and it's also a resource that's high in these polyunsaturated fatty acids. So it would have been another really great resource for these hunter-gatherers to have eaten. And I don't know if any of you guys are like me, but I love a really good seaweed salad. But as far as I know, and other people here attending can correct me on this, I honestly cannot think of, at least way back in time, any kind of even proxy evidence of linking people eating those foods. And again, it comes back, I think, to this idea of visibility. Something like that. It's hard enough to track plant use back in time. One of our co-directors of the P5 project, Dr. Renee Esteban, is actually attending. She's one of the listeners right now, and she's coordinating all of our plant proxy research. And she's just done a magnificent job of coordinating all of that work, but it's very difficult to try and reconstruct, not only the paleoclimates and the paleo environments, but also people's past use of those plants. Not to mention, to boot, if you added on a marine algae and seaweed, certainly that kind of stuff would have been brought in, even if it was just accidentally carried in on, say, the byssus of a mussel or something like that. But we've not seen any direct evidence that, for example, and I'm now speaking specifically about Impondoland, of that kind of a consumption. But honestly, I bet it did happen.
Darren?
Hi again. I was just wondering if iodine could be something to look at, but no. My question was, Erich, I completely agree with you 100% to look up at about the 10-kilometer coastal zone and not just the intertidal. I mean, one of the things that we have to remember when we talk about variability, and it can happen very often with sea level change, is that a rocky coastline, which is invariably most of the stuff we find in terms of shellfish are from rocky coasts rather than sandy, but a drop in sea level will turn a zone which was rocky into sandy, and all of a sudden what you've got is the corridor, but no longer really those immediate intertidal resources to collect, and vice versa. So that variability is one which is going to be very difficult to do. The other thing I think is useful to remember, that even though it is that this corridor, it is also a very narrow corridor, which means that whenever you get significantly and increasingly large numbers of people who, for whatever reason, are now no longer moving but are sitting around, it's very, very easy to very quickly overexploit that particular area. And that could be also a reason why these things disappear from the record, because it becomes really too much of a hassle. It's too far away to actually go and actually get them somewhere else, or you just consume them wherever else you're getting them. So you can start to see that drop in evidence coming, because people are becoming more settled, more sedentary, and that overexploitation, it's a very, very narrow fringe. It's very, very easily overexploited, especially as Curtis mentioned before, it's not a productive area. So it's something that you can graze on, but you aren't going to be able to live off it for any length of time. So yeah, unless you move. Sorry, that's it. Thanks.
Thank you, Darren.
Again, coming back to Impondoland, right now that coastline there, it's very rugged. You got these very tall cliffs, 30, 40 meters high, and in some cases they're completely impassable. And what we actually see is that there's evidence of significant movement, for example, during the last glacial maximum. And that's probably because when those sea levels moved out, even just a couple of kilometers, it created some access routes that are not available now. And so, yeah, again, coming back to this idea that Curtis proposed, is that really, I think the hardest part of this, other than just collecting environmental and whatever data from around Africa, modern day data, would actually be trying to figure out the configuration of those continental margins, because they would have certainly provided access routes. So, for example, in Impondoland, during the last glacial maximum, those coastlines moved out about 8 kilometers. And what it in fact did is it opened up a straight shot between our site at Waterfall Bluff and a site a couple of kilometers south, where we think that they were probably collecting lithic raw materials, hornfels. Now today you have to go up and around, and it's a pain to get at. But if the sea levels were moved out, you could have seen the site. You can see the site from, can see between the two of them, and you can just walk straight shot. So there's a possibility that during various times in the past, it would have actually been much easier to get to and from those places.
Great, thank you so much, Erich. So while for some of us is morning on a Friday, for some of us is the time for a true drink on the end of a end of a week. So thank you all for your participation. And before we say goodbye, I'm inviting you to already register for the next ICArEHB Dialogue. It's scheduled for the 18th of June at 4 p.m. And we'll tackle the topic of origins of stone tool. And if I can manage to share my screen, you will see who our guests are. Can you guys see that? Or are you seeing my wrong screen? Do you see anyone has to say yeah?
Yeah.
Okay. So our next guests are David Brown and João Marreiros for a dialogue about the origins of stone tool use. And you can already go to our website and register. So again, thank you all. A special thank you to Klervia and to Erich.